All?
Yes.
For centuries, matching Janam Patris, Kundlis or the natal charts have been an important aspect in the matchmaking process in India. But today's youth are opting for all different kind of matchmaking. Indians today are increasingly matching their genetic profiles before saying yes to their match. Let me take you all some few years back where we all at one point of time wanted to be as intelligent as late Rakesh Jhunjhunwala or as strong as the great Khalil or as smart as the ACP Pradyuman to maintain the level of professional skepticism throughout our audit. Was it possible to inherit the same level of intelligence or the strength level or the smartness back then?
No.
No.
Is it possible now?
No.
No.
No. Yes. Well, where the future is all about technology, this is possible. And this technique is known as human gene editing. But along with the technology comes its risks and issues. And so therefore, this technique also has some of the ethical concerns. A very fantabulous morning, ladies and gentlemen. It gives me an immense pleasure to welcome you all. My sincerest gratitude extends to all our dignitaries, jury members, members of the committee, branch and all my dear professional colleagues. Today, I am Srivastava. The subject matter on which I shall be sharing my thoughts and my presentation upon is ethical issues in human gene editing. So, these are some of the aspects on which I am going to talk about for the next few minutes. Before discussing about the ethical issues in human gene editing, it is very imperative for us to understand what actually human gene editing is all about. Gene editing or genome editing is a group of technologies that gives the scientists an ability to change an organism's DNA. These technologies, ladies and gentlemen, it allows the genetic material to be added, removed or altered at the particular locations in the genome. What does this genome means? So genome is the entire set of DNA instructions which are found in the cell. It basically contains all the information needed for an individual to develop and function. The first CRISPR technology was developed in late 1900s. More recently, if I talk about, that in 2009, a new editing tool called CRISPR-Cas9 has developed, which has made it easier than ever to edit the DNA and made the work of scientists very easy. Those who use this technology now uses CRISPR. Now what is this CRISPR? CRISPR is clustered, regularly interspaced, short palindromic repeats. It basically was identified and discovered in the genome of E. coli by the Japanese researchers in 1987. CRISPR-Cas9 was originally introduced or originally identified as a naturally genome editing system in bacteria. When infected with virus, bacteria capture these small pieces of virus's DNA and they insert them into their own DNA in a particular pattern that creates a segment called as CRISPR arrays. Now these CRISPR arrays, ladies and gentlemen, it allows the bacteria to remember the viruses or the closely related ones. So when virus attacks again to the bacteria releases the RNA segments from those CRISPR arrays that attaches to specific parts of the virus's DNA, and then the bacteria, ladies and gentlemen, it releases or uses the Cas9 or some other similar enzymes to cut the DNA apart, which disables the virus. Researchers adopted this genome editing system to edit the DNA. Genome editing is of great interest nowadays because it holds the promise to prevent the more complex disease such as cancer, heart disease, mental illness and HIV infections, to name a few. The categories of gene therapies. If I talk about the categories of gene therapies, there are broadly two categories. The first is somatic And the second is germline gene therapy. Somatic gene therapy, ladies and gentlemen, it affects only the patient being treated and some of his or her cells. Whereas the germline gene therapy, it affects all the cells of an organism and including the sperms and eggs. And so is passed on to the future generations, the possible consequences of which are difficult to predict. If I talk about the current scenario, ladies and gentlemen, there are over 2,000 gene therapies in development worldwide. And the global gene therapy market is expected to increase from $5.33 billion in 2022 to $19.88 billion by 2027. There is no doubt that gene technology is attracting many, but owing to its unprecedented and far-reaching consequences, there is a serious concerned dialogue that is to be required over its ethical and societal implications. Apart from its general awareness and apart from its general apprehensions, there are some of the major ethical issues. Let us see some of those. Off-target mutations. Off-target mutation is one of the major concerns which has been raised by the CRISPR-Cas9 technology. Because larger genomes can have many identical sites for cleavage, and hence CRISPR-Cas9 can cut those sequences which are not intended. And such technology can lead to abnormal cell transformations or even cell death. The second important and critical ethical issue is genetic enhancement. For safety reasons, ladies and gentlemen, genetic enhancement is banned in germline therapies. Therapy, the consequence of which is passed to the next generations. For that therapy, genetic enhancement is banned. But, however, CRISPR-Cas9 may allow the modifications in the DNA of somatic cells. For instance, one can improve the health of the sportsman. Genetic diseases which are caused by the mutations in DNA can theoretically be corrected by using germline technologies, but its consequences can be passed on to future generations, and questions therefore are raised on this novel change in the human genome pool. The very third is that this technology might not be accessible to all. It may so happen that this technology is only accessible to wealthy people, and this may further increase the disparities between the communities and in access to healthcare and other communities. The important and critical issue is privacy. Privacy, as defined by the ANA Code of Ethics 2001, involves the right of an individual to protect his or her own body, rights, and personal information. Now, it arises the ethical dilemma for the nurses, for the doctors, because many a time, patient does not want to share his personal information about the genetic information with their family members. For instance, a woman who has been tested positive for a cancer is not willing to share that information with their family members, which increases their chance of carrying the same genetic disease with them or might be to their future descendants. Privacy. No informed consent. It may so happen that it is impossible to obtain the consent in germline therapies because the patient being affected is the embryos and the future children. So how can you obtain the consent from them regarding the germline therapies? To conclude, ladies and gentlemen, I would like to say that CRISPR is no more a word of scientific community. It has crossed the barriers of the global market and has entered into the fields of pharmaceutical and biotech business segments. Yes, with such hype and frenzy, it might not be easy to confine this technology only for the non-medical use. However, the debates, the discussions, the worldwide awareness will definitely bring this technology to the new level. As in the 21st century is not digital, it's biological. That's all, folks, ladies and gentlemen. Thank you so much for being such a